Okay, so here we are. We are going to go through the second half of chapter 14, where we are now going to focus on, or learn about chloroplasts and how they are the center of energy production in a very different way than mitochondria. They are actually the site of photosynthesis in plants. So we are going to compare and contrast mitochondria and chloroplasts as we go through this part of our lecture. So last time we talked a lot about mitochondria and how it provides energy in the form of ATP to our cells by taking those pyruvates and acetyl-CoAs and then breaking them apart to produce your NADH, FADH, and GTP molecules in the first stage that are then these activated carrier molecules then take those electrons and take them onto the electron transport chain where they can then be utilized for ATP production. And we also talked about how electron transport chain is organized within that inner matrix membrane or inner membrane of the mitochondria with the matrix containing all the enzymes needed for this this production of ATP, and how there is this stepwise manner in which several enzymes work together to harness the most energy out of each glucose molecule that we are breaking down in the process. So the NADH and FADH2 which have those electrons in their activated states, pass them over to the next next electron carrier until the end product, which is your ATP production in ADP synthase. And this process takes partly through pumping and shuttling of protons as electrons are passed and transferred to the next electron carrier at each step, whether it be a ubiquinone molecule or a cytochrome C molecule, and then finally to your the final electron carrier in the form of oxygen, which which we have talked about, which then finally transfers it into a state where you can get your ATP synthase to start rotating with the production with the movement of protons back into the matrix from that intermembrane space where they have been shuttled throughout this process. So today we are going to shift our energy a little bit differently, and we are now going to look at how chloroplasts in plants, which are the site of photosynthesis, also resemble mitochondria in many ways. However, their function is going to be to utilize that, to capture and utilize light energy so that they can produce those molecules that are needed in order for ATP production down the road. So going into it right away, let's talk about what exactly is produced by photosynthesis. So if you look at the various steps of photosynthesis, you can actually see that any organic material that we can possibly require in a living cell, or that is possibly required by a living organism, can be produced at various steps throughout the process of photosynthesis, or as a result of the products of photosynthesis that are then further broken down to get those other macromolecules building blocks. So photosynthesis is essentially a series of light-driven reactions where the light is coming from that energy that is harnessed from sunlight or another light source into the chloroplast, and they are used to create molecules of some sort from atmospheric carbon dioxide. So in this case, again, you are using water. Water is essential for all living things, and you can see that at every single step. We've talked about how much water is required to actually get these processes moving, whether it be one of the metabolic pathways or breakdown of macromolecules or generation of nucleotide and DNA and or RNA, every single process in our system, a majority of them require input of water in some way, shape, or form as bonds are getting built or broken. So again, in this case, you are using water molecules that are broken apart. So through the process of hydrolysis, while you generate your glucose molecules, you also are producing this oxygen as well. This is primarily performed by plants, algae, and certain cyanobacteria, and it generates first in it generates as well as it's going to consume ATP and NADH as their energy source. So on one side, it's going to be consuming it in order to produce some of these macromolecules, and then it's going to break them down again and get its own energy needs covered through production of ATP and NADPH. So looking at that, we can look at when these photosynthetic organisms started out, and we can see that they were formed very early on in our Earth's environment as our atmosphere got more oxygen within it, and as more light was coming to the Earth's surface that could be harnessed by these organisms to utilize for energy production. The original photosynthetic organism was probably a bacteria that over time then changed as it evolved into the various other types of organisms and creatures that you see that have this ability. And this allowed them to utilize the oxygen in Earth's atmosphere and convert into a aerobic organism, being able to utilize that oxygen for their production of energy needs instead of the anaerobic environment they were living in before there was enough oxygen in the Earth's atmosphere. So chloroplast, let's look at their structure and how they are organized. You will notice right away that they have some things that are very similar to our mitochondrial structure. So here you again have double membranes. So you have an outer membrane that is facing the cytoplasm or the cytosol. And this outer membrane is again, just like a mitochondria, extremely permeable to the cytosolic factors. So a lot more molecules can come in and out of this than a typical plasma membrane. So this is a more permeable membrane than a plasma membrane. In addition, you again have the inner membrane that is going to be a lot more restrictive, just like the inner membrane of the mitochondria, which is a lot more restrictive in what it allows in or out. And so those two are very similar to how you would see it in a mitochondria as well. However, right away you can notice that this inner membrane is not invaginating or creating those cristae structures that we saw in mitochondria that made that, large space inside the center, which was the matrix. However, still, this region inside the inner membrane, the stroma, is actually very similar in some ways to that mitochondrial space and contains a lot of, it is again a very enzyme-rich kind of environment. The difference here, however, is that while it does contain some of those same kind of metabolic enzymes and similar to what you had in the mitochondria, it does not contain those enzymes related to ATP production in the form of the electron transport chain. All those materials, all those enzymes, all those receptors have actually moved into this thylakoid membrane, which is this third kind of set of membrane that is within the chloroplast that was missing in the mitochondria. So the thylakoid membrane is again wrapped around itself, similar to how you saw the inner membrane of in the mitochondria, which created that matrix area. However, this one is kind of folded over on top of each other to create these flattened stacks. And these flattened stacks are called grana, that are, well, the discs are called thylakoid. Well, the discs are made of thylakoid membranes, and then individual discs in here are what we call thylakoids, and then when you stack them up on top of each other, you get what we call a grana. And these spaces, these little stacks, are the site for the electron transport chain, photosynthetic, ability to capture the light from sunlight so that they can run photosynthesis. And that all this compartment that is inside of it is where all those enzymes are going to reside, and these membranes are going to be where those works are going to get done. So this is something a little bit different than mitochondria, where you only had the two membranes and the second membrane had created that third space. So here, the thylakoid membrane contains all those enzymes that are going through and performing those important functions related to photosynthesis as well as electron transport chain. So we're going to look at them more in detail in a minute and look at exactly what's going on at the molecular level inside. So again, in a photosynthetic cell, you're going to see your cell membrane and a outer cell wall. You will see your large vacuoles that are maintaining that cell's kind of water, equilibrium. And then you will have all these chloroplasts. So what we are looking at here is inside one of these chloroplasts and looking at what it is that is making up that space. So inside, again, you have the stroma, which is kind of like the matrix, but then inside that space, you have that third membrane in the shape of thylakoid membrane that is creating the thylakoids and stacked together in the form of grana. It's kind of like a stack of pancakes, where each of the pancake is a thylakoid, essentially. And that's where you're going to see a lot of these reactions happening that we are going to talk about. So now the photosynthesis in a chloroplast happens in two stages, and they are typically, labeled light dependent and light independent reactions, or you may have heard of the term light versus dark reactions as well. The light reactions, obviously, just like the name implies, require energy stored into these thylakoids from the light that are then used to create the macromolecules needed in the form of glucose and to release the oxygen into the atmosphere as a byproduct. So that particular portion is light reactions or light dependent reactions. The molecules that are then taken in are, the thylakoid membranes then take in that light that they have taken in, and they produce ATP and NADPH from it that is used in the second part of the cycle in the light independent reactions to give you the final sugar molecules or the amino acids and the fatty acids. So the first part, this you can think about these little granas or the thylakoids as the, as kind of the energy center or, light or energy plant that provides the electricity for the rest of the factory to do the work it needs to do. So this portion, the light reactions are only providing the energy needed to run the actual factory, the second part of the reactions, which is where you're making all the macromolecules needed for that organism to survive. So this one is run with water. It's a hydro plant, right? So just like a hydroelectric plant or a dam that is used to run an electric plant, this one is also run with energy from water and energy from light taken in and breaking it down to produce ATP and NADPH that is then utilized in the second step in the presence of carbon dioxide, using carbon dioxide from atmosphere to produce your macromolecules needed for survival, including sugar, amino acids, and fatty acids. So quite a cool, efficient factory in there. In this case, light that you are taking in from the sun is going to behave as essentially a stream of photons coming into, they're taken as streams of photons taken into the thylakoid membranes. And each photon basically contains what we would call a quantum of energy. It's just basically a small unit of energy. These are going to be dependent, the amount of energy or the amount of quantums per photon that you are getting in is going to be related to the wavelength of the light that it is taking from that light stream. And so, different types of light, different colors of light are going to provide different amounts of energy per for each use that it gets. So the wavelength of a photon in general is inversely proportional to the quantum of energy. So the shorter wavelengths are going to provide higher energy, higher quantums of energy, and the longer wavelengths are going to provide less energy per unit. So blue light, for example, has a shorter wavelength than red light, so it's going to give a lot more energy, provide a lot more energy per unit than red light would. These photons are absorbed by specific pigments inside those chloroplasts, and those are going to be then utilized and used as energy is transferred to electrons within there. So the pigments and the color that the pigments have to begin with dictate what type of wavelength they can utilize, whether they can utilize the short wavelength or long wavelength or a combination of those wavelengths. And we will see that in a couple of slides as well. So once they absorb these photons, they essentially use that to excite electrons. And those electrons, when excited from their ground state into this excited states, this process is called photoexcitation. Basically, they are getting excited using that light energy that is being taken in the form of photons, and that quantum energy is getting transferred to them. So sunlight specifically is usually absorbed majority of the times by chlorophyll. Chlorophyll is one type of pigment molecule that you can have that can absorb this light, right? This is the one, chlorophyll A and chlorophyll B. Those are the ones that are most commonly found in plants and in photosynthetic organisms. But that's not the only pigment that's available, and that's not the only, the light range that they can absorb is not just the ones that you get from chlorophyll. This is just the one that you most commonly see. The chlorophyll molecule contains this porphyrin ring on one side that is stabilized with magnesium ions. So again, you're looking at metal ions getting incorporated into these molecules to stabilize the structure or to enhance their function. Just like you saw the copper ions and the iron ions in mitochondria in the electron transport chain to utilize for the excitation of electrons and transfer of electron energy there as well. So the light is absorbed by the electrons in these rings that are present inside this ring, and in that state, once they're excited, they can then store that energy. It's like a solar cell, and then utilize that energy later on. The tail is just a hydrophobic region. If you look at that, that's essentially just a carbon-hydrogen chain, and it is embedded within the thylakoid membrane because of this hydrophobic nature of this tail. Now, if you look at the wavelengths that the chlorophyll can absorb, that they can utilize, you will notice that there are two peaks within this spectrum. So this is just looking at visible light going from your, lavender, or not lavender, but, indigo and violets all the way up to your reds. The blue range as well as the red range are both absorbed by chlorophylls, which is great because that means that a bunch of the spectrum is getting used by this particular molecule. However, you see there's a large portion of this spectrum that is not utilized by this as well. So the only energy that they are able to harness is the one that is seen in the red light range and the red wavelengths and the blue wavelengths. And you'd see a clear peak. That is the point at which the highest amount of that absorbance is being taken in by those chlorophyll molecules. So highest amount of that energy is getting utilized at these two peaks. So given a spectrum like this, you should be able to tell what wavelengths that particular pigment molecule is able to utilize the best for photosynthesis or for energy storage needs. And here you can see some of those other alternate, minor pigments that you will also find in plants, as well as other photosynthetic organisms. Now, pigments, just because a plant looks green doesn't mean it contains just green pigment. You would have, if you did GB1 here, you would have done this chromatography experiment with spinach, ground up spinach, where you would have actually seen that in action, that you can see all these other accessory pigments present in there as well, or a large range of them present in there as well. They're just present at a lot lower concentration. So if you are looking again at this particular graph, you can see that chlorophyll A and B, majority of that takes in just the red and the blue lights. However, all these other accessory pigments that are available, whether it is phycocyanin or carotenoids, they can take energy from other ranges in that visible light spectrum. So having some of those other accessory pigments allow the plant, allow those photosynthetic organisms to take in more of the spectrum to utilize it for energy use. Okay? So one of the things that you might wonder is that, well, if you think about how light is absorbed versus reflected, we know that if you have some object that is white, all the light is getting reflected back to you, or majority of the light is getting reflected back to you. And if you have something that is black, everything, all the lights, the entire spectrum is getting absorbed in. So maybe you want to wonder about and think about a little bit, why is it that plants are not black? Because if they were black, they could utilize the entire spectrum and they would be able to absorb and utilize all the energy from the sun instead of just utilizing a portion of it. I think that would be a great critical analysis question on the exam. Or some derivative of it, who knows? Okay, so let's think about, let's just kind of think about what they are doing as they are getting excited, as they're absorbing that energy. So once the chlorophyll molecules gets energy from these photons that they are taking in from the different wavelengths that they are able to absorb, they then take those energetic molecules, the excited chlorophyll molecules, and funnel that energy into a reaction center that we will see in a second. Now, anytime an electron is excited state, it is usually unstable. It is going to be unstable. And it has three different options on how it can get back to a more normalized or ground state of just existence. One of them would be for that molecule to release its energy by fluorescing. And so in that case, it's going to release that energy that it's stored in that excited stage, either as light or as heat. Another way for it would be to pass that on to some other electron so it gets back to its normal state and gets somebody else excited. And that would be what we would call a resonance transfer, or energy of excited. One electron is getting transferred to a nearby another electron. And finally, that excited electron can be removed from the pigment, stripped away from that original molecule altogether, and then captured by one of those energy carriers to activate them, like your NADH, like the ATP, ADP, when it, whatever it is that is going to be that intermediate carrier or the final carrier. So in this case, again, you would want to see how that works, right? So a pigment molecule in its ground state, it's in happy-go-lucky state, it's a very stable, it's just going about its business. A photon strikes it and transfers some of that photon energy to it and moves it up into this excited state where it's now unstable. So if you were to look at it a little bit closer, you will notice that that meant that when it absorbed that quantum of energy, it shifted it in its orbital space from its own electron to its from its own orbit into a higher orbit, higher energy orbital space. And that movement now makes it unstable because now it has to do more work and it doesn't know what to do with all this energy, and it needs to go back to its ground state where it likes to hang out, especially since that inner orbit is now not at its happiest state itself because it's not totally filled. So as we talked about, it has those three options to go back. The most common option for these molecules is to go through what we call a photochemical reduction, where a relatively stable higher energy molecule orbit is formed where this electron can be housed while it is in its excited state. So what we see inside the inside our chloroplast, and if you were to look inside those thylakoid membranes, in between that thylakoid space, right? So you have the membrane on the top and the bottom. And then if you were to look at that membrane close up, you will see that you have your chlorophyll molecules in the reaction center and one side. They are that's one of the places that you will have them. Then in between, you have these stacks of chlorophyll molecules on either side of the reaction center. And this entire area is what we call a photosystem. So you have a reaction center with a special pair of chlorophylls, which we'll talk about in a second. And then you have these little light harvesting units within these little, you can call them little batteries. Each one of them containing chlorophyll molecules all stacked inside of them. When light hits one of these chlorophyll molecules, it excites those, it excites the electrons within it. It gets them into an excited state. That essentially gets transferred from one chlorophyll molecule to the other until they reach the special pair of chlorophyll molecule in the reaction center, where that energy could then be stored and processed. So these individual stacks of chlorophylls are what we call light harvesting antenna complexes. So these are just a bunch of chlorophylls joined together inside, not joined as in physically joined, but just kind of stacked together inside these harvesting antenna complexes, where they can be used to transfer energy from one place to the other. So inside that actual reaction center, what's so special about that system itself? The special pair transfer inside the chlorophyll that is in the reaction center, those chlorophyll molecules are going to be special in the sense that they can now transfer these electrons to an electron transport chain. So while the others were just transferring it to one place to another, the special pair, when it transfers the electrons, it transfers to a mobile electron carrier that can then move it into the electron transport chain in the reaction center. So it does not have any more chlorophylls that are attached to it, but rather it has a mobile electron carrier that can get activated and transferred over. This system is made possible partly because as the electron leaves the special pair, it makes these chlorophyll molecules positively charged. So they would be more likely to accept electrons again, right? So this system is basically made with that special pair having that positive charge without an extra electron on them. And that electron then moves on to your mobile electron carrier stage, which then can transport it or move it itself physically into the electron transport chain. Now, in the process, your reaction center and the special pair, once it loses that electron, it will set it back to its reset. It kind of resets it back to its native state, where now a new electron can come in from the chlorophyll molecules in those charged antennas to allow this process to continue to function. So there are different photosystems within your thylakoid membranes, and so this first system sets essentially provides these electrons into the electron transport chain. And then the second system, the photosystems 2, is what is going to set the proton gradient for ATP synthesis, just like we saw before. So here you have your thylakoid membrane that has the reaction center and the antenna complex on either side. Light energy is hitting the chloroplast, the chlorophylls inside these antenna complexes that are then transferring them into the reaction center, where then the electron is released and goes onto a plastoquinone molecule in this state. This is equivalent to the ubiquinone molecule in your mitochondria. And that plastoquinone molecule is that intermediate carrier of electron. Now, this molecule is going to go into the cytochrome B6F complex. This is, again, if you go back and compare it to the electron transport chain in a mitochondria, you had the cytochrome C complex, and then you broke it down. First you had the cytochrome C taking the electron, and then you had it getting reduced again to oxidized again to get your electrons transferred the second time to produce that proton gradient at each step. Here, you have only one complex in the center, and that's the cytochrome B6F complex. And this complex is what is going to produce the proton gradient by, as it takes in those plastoquinone and separates those electrons, it is going to shuttle hydrogen ions or protons outside the thylakoid space into the stroma. That increases that the amount of those protons in that space, in the thylakoid space, and allows for it to then get shuttled back into the ADP synthase with the proton gradient, so that, again, similarly, ADP can be made into an ATP for as these protons are coming in. So the first part of the system, remember we talked about how it is going to produce essentially some type of a carrier molecule, and this was your photosystem 1, which we were talking about. Looking at it a little bit closer, let's see what it is that it was generating. So you had the light hitting your antenna system, you had your special chlorophylls in the center, in the reaction center. The electrons that are made from there are actually transferred first to a ferredoxin intermediate carrier that binds to a ferredoxin NADP plus in reductase molecule to produce NADPH. So this one, instead of NADH in mitochondria, you're getting NADPH instead. It's a NADP plus ion that combines with a proton to produce NADPH at the first step. And that NADPH is what is then moving this process to the plastoquinone in the second step. Now this process, it requires, again, photolysis of water. So you're still using water, you're creating hydrolysis, you have to do hydrolysis in order for this reaction to run. It cannot run on its own. So that water is getting utilized and getting broken down by light in this case, the light energy in this case. So it is a photolysis of water that replaces the lost electrons in your system, in the photosystem 2, as you get to that second step. The manganese cluster inside those chlorophylls aids in this process to split the water inside the special chlorophyll molecules so that it can be split to produce oxygen that is released back into the atmosphere, and 4 hydrogen ions that are then taken again into that thylakoid space so that it can be used utilized by your ATP synthase at a later time. So here you have your thylakoid space in the top, and you have the stroma, the outer space on the outside. And so your ATP is going to get produced towards the stromal space, and all these reactions are happening inside the thylakoid space on the top. And you can see a little bit of up-close reaction here. So you have your manganese clusters that are going to help aid the breakdown of this water to produce 4 hydrogen ions, 4 protons, and oxygen that's released back. The electrons are utilized, are moved into the special pair of chlorophylls that then plaster it onto that plastoquinone molecule that can then enter photosystem 2 reaction. So here you can see those different types of molecules that are in the photosystem 2 versus photosystem 1. So in the first step here, well, both of those requiring light for energy production or energy storage. So here is one part of your molecule, of your system. This is the first part in photosystem 2, where you are taking the light energy and you are using the water splitting enzyme, which requires magnesium, to break down the water into oxygen and protons. And this is going to allow those special chlorophylls to release that electron onto a plastoquinone that can then go into a cytochrome B6 complex to shuttle those protons out into the into the thylakoid space. You then also in the process are making a plastocyanin molecule that can also be utilized with light to work on the ferredoxins to allow them to bind to FNR and change NADP plus to NADPH, and utilizing some of those protons that are in the stromal space. And this is your photosystem 1. And then finally, you have the proton gradient that is going to run your ATP synthase, just like it did in the mitochondria, by shuttling in the protons back into the stroma, where it is going to rotate the ATP synthase and convert ADP, use that energy to convert ADP into ATP. So the photosystem coupling allows this process to be more efficient. It allows more energy to be taken from the electrons by exciting them enough so that they can be used to make NADPH. So just without the light energy getting added into it, you wouldn't have enough excitation power within these electrons to create NADPH. So the photosystem 2, which comes first, it's a little bit of a misnomer in that way, is what is using water for its energy source to process the first part of the system, and the electrons that are charged in this process produce these electrons that are then converted to the plastoquinone molecules. And that plastoquinone is then going to be moving that through the cytochrome B6 complex to generate the proton gradient by shuttling those protons into the thylakoid space. And then as it gets through the cytochrome B6 complex, the plastocyanin that is a result of that will get further excited inside the photosystem 1 through, again, taking in the light and using that light to excite these electrons that can then be transferred to the ferredoxin, and with the help of FNR convert NADP plus to NADPH, which requires some proton as well. So the light energy that you're producing in these two systems, in the first case using the electron transport chain, and then in the second space just using the light energy itself, is in the form of ATP and NADPH. And now you can see, by the way, that in photosystem 2, where you were using electron transport chain for energy production, you actually produced that higher energy molecule, the ATP. While if you were to use just the light itself to excite these electrons and convert it into energy, you got the lower quality energy molecule, which is the NADPH. However, both these molecules are helpful, obviously, for our cells to get to utilize for macromolecule production. So I'm going to stop here for this particular part of this lecture, and I will be making a second video where we will talk about carbon fixation and more about photosynthesis itself. So in this first video, you have learned about how chloroplasts work, similar to how mitochondria work, and how they are different in production of energy, right? So this was just looking at that. And in the next part, we will talk about how photosynthesis works in these same space and how you get your actual macromolecule production.